Hi, I just want to spend a few minutes walking you through a single experiment in LabSmart. We're going to take a look at cell structure 2, diffusion and osmosis, and we'll take a look specifically at the diffusion through a semi-solid lab. So just to get us oriented, the lady over in the left is the coach. The coach has several functions, and I'll review those as we go through. Over here on the right hand side of the screen, we have the review of core concepts area, then the diffusion experiments, and the osmosis experiments over here on the far right. The first place we're going to look is the review of core concepts here in yellow. Review of core concepts is also known as the leveling. So the goal of leveling is to make sure that all the students who go into the simulators in the diffusion and osmosis experiments are at about the same level in terms of their content. So it'll get them all up to the same speed in terms of the basic concepts they need to be successful in the diffusion and the osmosis experiments. So let's take a look at review of core concepts. For those of you that are familiar with LearnSmart, there'll be elements of the leveling review of core concepts that look very LearnSmart-esque. So the first role of the coach is to help the student understand some basic content. So once LabSmart moves out of beta, all of the coach speak here in the bubbles will be fully narrated and available in audio. So here, the coach is explaining the content over in the slide on the right hand side. So the student's receiving a little bit of content. When they've listened to the coach and they've looked at the slide, they click continue, and then they'll enter a LearnSmart kind of mode. And so the student's being presented a question or a probe around a particular learning objective. To show you some of the features of the leveling, I'm going to get some of the questions wrong and some of the questions right, so just bear with me. The solvent in biological solutions such as cytoplasm and blood is, and I'm going to get this wrong and say oxygen. So when I get a question wrong, the system records that I didn't understand that topic or that learning objective, and the coach is now going to explain to me the concept that I didn't understand. So here, I was having a problem understanding solutes, solvents, and solutions. So the coach, again on the left, is going to explain the concept to me, and when I've listened to her and I reviewed the slide, I'll be able to continue by clicking on the I get it button. In a solution, there is always more of the blank compared to the blank. So I'm going to say I know it, and I'm going to get some of these correct now. In a solution, there is always more of the solvent compared to the solute. So I'm going to have to get a few questions right before I'm allowed to move on to the next learning objective. Air is a solution of gases. It is composed of 1% water vapor, 0.04% carbon dioxide, 21% oxygen, 78% nitrogen. Which of the gases is the solvent? Nitrogen. A solution is a mixture of a solute dissolved in another substance known as solvent. The solvent in biological solutions such as cytoplasm and blood is water. Which has more solvent, a 10% sugar solution or a 20% sugar solution? The concentration gradient exists in a solution when the concentration within the solution is uneven. So when I've convinced the system that I understand or have mastered the learning objectives, the system's going to tell me that it's going to save me some time and it's going to let me move on without having to go through the remediation. For those A-type students who really want to see the remediation, they can see the slides and listen to the coach anyway. I'm just going to continue. And so I would now move on to another topic in the leveling. So there are two key features here of the leveling. The first one is that when a student needs help, they receive the help. They get the help when they get the questions incorrect. But if they demonstrate that they understand the material, then they will save time and they don't have to go through all the remediation. And this way, all of the students that go through the leveling should arrive at the end of the leveling at about the same type of content knowledge. Okay, let's go back to the menu and we can go into the diffusion through a semi-solid experiment. So I click on main menu and we're back at the beginning screen. I'm going to come into the diffusion set of labs, and here you can see there are some more core concepts review, and this behaves exactly the same as the leveling I just described. Then there are 4 experiments and then a final review. So we're going to go into diffusion through a semi-solid real quick. And I'm going to go through this pretty quickly. Okay, so here is the coach performing her second role. Her first role is to help the students with content. Here, she's guiding the students through what they should do in the simulator. So she's going to set up the scenario for the students. And the student scenario here is that the student has to determine how diffusion speed depends on concentration gradient. So the coach will explain what the student needs to try and do. There'll be a short tutorial that the student can watch which will show them how to use the simulator. This way, when the student goes into the simulator, they're not completely confused and lost. So they'll learn how to use the notebook, how to take screenshots, and how to manipulate the pieces of equipment that they've been given. So when the student has viewed the video, they click on continue and they're into the lab. Okay, so the first part of the lab requires that the student decide on a hypothesis and then an experimental strategy. So let's pick a hypothesis. There's no right or wrong hypothesis, the student just has to pick one. A student that knows what they're doing will probably pick the correct hypothesis. So, I'm just going to pick concentration has no effect on diffusion rate, which is not true. I then have to pick a strategy to test my hypothesis. And this is where the system becomes interesting. The system's recorded the hypothesis I chose, and it will record the strategy I choose to test my hypothesis. And if my strategy does not test my hypothesis, it will record that I've made a poor choice. And later, I can come back and try a different strategy. Okay, so what strategy will I use to test my hypothesis? I'm going to say that I will use an increasing number of crystals on each subsequent plate. So I now come into the simulator. The coach is up here in the top left-hand corner, and she'll provide a little guidance if I need it. She's not going to tell me exactly what to do or when to do it, but if I make a critical error, she'll give me a little help. And so that's her third role. Stopping the student from progressing if they're going to make a very serious or critical error. So I've got 3 plates. I can pick up crystals over here on the right, and I can drop them on the plate. Now, I said that I was going to use an increasing number of crystals on subsequent plates, but I'm not going to do that. I'm going to put 1 on plate 1, 1 on plate 2, and 1 on plate 3. So the system knows what my hypothesis was, what I said my strategy would be, and it will now record what I actually do. So if there's any lack of alignment between those 3 things, the system will know that I've got a problem understanding the process of science. So I can put my little forceps down up here, the little stopwatch has appeared, and I can accelerate time. And so as I accelerate time, diffusion of the crystals occurs in each of the plates. I can pick up the little ruler, I can place it over the diffused crystal of dye, I can zoom in on the plate, and I can make a measurement. And the system knows that I'm doing all these things, so it knows I picked up the ruler, it knows I've zoomed in on the plate. I can take a snapshot of that screen. I'm going to say that's 14 millimeters. I zoom out, and I can measure the other 2 plates. I'm not going to do that just for the interest of time. I can open my little notebook here, and I can say that at 60 minutes, I have 14 millimeters, 14 millimeters, and 14 millimeters. And obviously the system knows what the actual diameter is here. So if the student just makes a number up and puts that in there, the system will know if the student is close enough to the actual diameter of the diffusion of the dye. I could take another measurement after an additional hour, and then I could put that in my lab notebooks. Now I'm not going to do that here. I'm going to try and make the system respond to me based on poor choices that I make. So the student can make as many measurements as they like here. They can use the little button up here to switch out the plates, and that will clear their data out, and they could do another experiment. So what you have here is a system where a student can experiment, but they're being kept in the confines of, let's say, a box. So if they try and step outside of the box and do something entirely crazy, they're not allowed to do that, but they can experiment within the confines of this box, and that's good for students as they need a little bit of structure. Okay, so when I as a student have done enough experimentation, I can click on the To Analysis button down here in the bottom right hand corner. And so you can see that my progress through the lab is tracked at the top here. So right now I'm in the experimental stage, and when I go into analysis, the coach is going to come up here real quick and say, look, you don't have enough measurements to move on. If you want to go on, that's up to you, but you haven't made enough measurements. She's not going to tell me exactly what to do, but she's going to say, look, you've probably got a problem here. You might want to make some more measurements. I'm going to say I don't care, and I'm going to move on to the analysis. So we come into the analysis, and the coach is going to give me a prompt here that I need to finish up this, and I need to create a graph. And so what I should be doing is, if I had enough time points, maybe 3 time points and 3 separate sets of measurements, I would be able to calculate the speed of diffusion down here at the bottom. And so the student actually has to do some calculations here, and the system knows what the correct diffusion speed should be based on the data the student collected. They're required to add a graph, and they can create different types of graph. Let me just create one type to show you what this would look like. So let's do time against distance, and I'll make a bar graph. This is a pretty reasonable graph to do. A few little bugs there that need fixing, but we'll get those fixed, and I can add that graph to my report. If I try and make a graph which is nonsensical, like if I tried to do time against diffusion speed, remember I didn't calculate diffusion speed, and I say I'd like to do a line graph, I should get an intervention from the coach here because I haven't calculated diffusion speed, so I can't plot that on my graph. So again, the coach is going to stop me and just give me a little prompt when I try and do something which is either a little bit silly or is kind of trying to step outside of this little confined box that I'm working in. So if I'm happy with my graphs and I'm happy with my data analysis, I can continue. The coach is telling me again that I haven't calculated the diffusion rate, so I haven't really done everything I'm meant to, but I'm going to be a typical poor student, and I'm going to move on anyway. The last thing she's going to ask me to do here in my analysis stage is to determine whether the data I collected, my results, actually support my hypothesis. And again, I'm going to say yes, they do, but they don't. And then I move on to this pre-report, and this shows me where I've got some problems and I need to go back. And so if I've got everything filled in green, then I've done okay. And where I've made errors, for example, I didn't use the appropriate materials. I said I would do one thing and I did something different, so I need to remediate on my use of my materials. I can go back into the simulator and I can make those corrections, or I can possibly answer some questions which would show that I understood the errors I made. And when the student is happy with their pre-report, they can go into their final report, and at this point they cannot go back, and this is what will go to the instructor. A lot of work is being done on the reports right now, so this will likely change. But the report will basically summarize what the student did, the equipment they had available, and it will put in their exact manipulations. And so if you remember, I put 1 crystal on each plate, and that's described here, and I time skipped forward to about 1 hour, and then I used the ruler, and so it's measured all of those things so the instructor can see exactly what I did. And then the screenshot that I took has been put into the report. So again, if the instructor needs to see that I actually did some measurements and I was accurate in my measurements, they can see that. And they've got my little graph in here, and if I decide that I've got too many graphs and I don't need that one, I can delete it. I've got my data table here, and so if I've taken 3 sets of data and decide that one of them isn't very good, I can again delete them using the big orange cross. And then at the end, if I'd gone through the discussion and conclusion stages, there would be some content down here at the bottom, but I didn't go through everything. I just wanted to show you what the simulator looked like and the student workflow. So I'll just come back to the main menu here, and we're back at the beginning. And you can see I've got a calculated grade here. I didn't do very well, and ultimately this grade would go to the instructor into Connect, and the student could print out a PDF of their report and turn that into their instructor, or they could load it up into a custom assignment in Connect. So that is a very quick walkthrough of a process of science lab, the diffusion through a semi-solid, which is part of the diffusion suite of labs, which is part of Cell Structure 2, Diffusion and Osmosis.